that's focusing on breast cancer in stromal versus epithelial cells. A little bit about why I chose this project. Cancer has always been a topic of my interest, and specifically breast cancer. I have done a few presentations on breast cancer previously, however, those were more clinical based and focused on pathogenic variants. This article and dataset were more of a molecular focus on the stromal and epithelial cells, which I thought was a different perspective and would be interesting to analyze. I also thought it would be a great way to further dive into the topic by applying different machine learning algorithms that we've learned in class to further understand what is happening. Additionally, cancer can begin in any cell in the body, so for that reason, it's critical for all scientists, researchers, and clinicians to understand the importance of the various types of cells and the environment that these cells reside in. Using machine learning algorithms, these cells can be studied and investigated to further understand disease progression. Moreover, these techniques can also be beneficial in evaluating gene expression and creating accurate models to advance cancer research. The purpose of this project was to find the significance of stromal and epithelial cells in normal and cancerous breast tissues using a variety of the machine learning techniques to create a model. The problem/challenge I decided to focus on was if we can determine if an individual has cancer based on gene expression levels. Based on the dataset, I predict that regardless of the cell type, epithelial or stromal, we will be able to predict if cancer is present. However, it is possible that expression will vary among features in the epithelial cells versus the stromal cells. So a little bit on the research. Next to skin cancer, breast cancer is the most type of common cancer among women. However, it can also affect men. There are two main types of breast cancer known as invasive ductal carcinoma and invasive lobular carcinoma, and there are three different parts of the breast that can be affected, which are the lobules, the ducts, and the connective tissue. Age, genetic mutations, reproductive history, and family history are all significant risk factors to the development and/or progression of breast cancer. The tumor microenvironment consists of many different components that typically enhance tumor progression. This includes blood vessels, immune cells, stromal cells, proteins, RNA, extracellular matrix, and more. Additionally, the importance of the tumor microenvironment lies in the fact that metastasis, the spread of cancerous cells to other organs in the body, is about 90% of the cause for death in patients with cancer. Therefore, evaluating the surrounding environment of the body's cells and where they reside is important to furthering cancer research and saving patients. The dataset that I chose is obtained from GEO, and its ID was GSE1797. It contains 66 samples and 22,278 genes with 3 different classes. The associated article was Molecular Signatures Suggest a Major Role for Stromal Cells in Development of Invasive Breast Cancer, and epithelial cells are cells that cover different surfaces inside and outside the body, including organs, while stromal cells that make up the connective tissue, connecting different organs and tissues together. Knowing the difference between these two cells is significant to understanding their role in cancer and where they might be found. So my testing methods included a few, which I will further explain in the next slide, but I have them listed here as PCA, which is Principal Component Analysis, Lazy Predict Model, Autoencoders, Random Forest Model, Confusion Matrix, Feature Significance, and one that I do not have listed is Decision Tree, but that is also one of my testing methods that I included. Principal Component Analysis, or PCA, is a method used to determine similarity between different groups seen by clusters of the groups within the plot. Lazy Predict Model is a method used to obtain a classification report that illustrates the accuracy of specific models, ultimately telling us which model is best for the data. Autoencoders are a method used to represent the most important or useful information from a dataset, representing a neural network which is trained according to the provided dataset. Random Forest Model and Confusion Matrix are two methods used to visualize and classify the data. Feature significance is a method used to determine which features or genes within the dataset are most significant based on the given model. Now for my results, the first result obtained was the PCA plot, which shows 26% explained variance. With the lower percentage of variance explained, we see that there is no clear distinction between the three groups, as the normal cells are scattered among the stromal and epithelial cells. Thus, further refining and analyzing needs to be done to obtain clear results. Next, a lazy predict model was done so that we could obtain a classification report which shows us the accuracy and time taken for various models. The first image shows numerous different models, while the second image shows the top 5 models. We can see that the decision tree classifier takes the least amount of time while having the greatest amount of accuracy and F1 score. ADA Boost classifier also reports similar accuracy and F1 score to decision tree classifier, but comparatively takes much longer. The next 3 models also show lower accuracy and F1 scores compared to the first 2 that were mentioned. These images show autoencoding with 100 features, which are the top 2 images, and 50 features for the bottom 2 images. This was done using the TAN-H optimizer, and with 100 features, we can see a 90.34% variance explained, with the test and train data at a mean absolute error of about 2.6. And additionally, in the PCA plot for 100 features, there's a clear distinction of the cancer stromal cells. Another optimizer, ReLU, was attempted to be used, however, the results were unclear. But with no clear separation happening in the second plot with 50 features, there is definitely more improvement that can be done with the optimizers and with this autoencoding, but that could also be done with future work and further analysis to continue this research and study. This is a confusion matrix which was done to help visualize and classify the data. From the model, we can see that there is misclassification within all 3 groups. Therefore, to create a better model, I decided to combine the cancer cell types and compare cancer versus normal. This resulted in a second confusion matrix, which I will show in the next 2 slides. Before the next confusion matrix, I have a decision tree, which shows the splitting of the cells when they're classified. So if a feature has less than or equal to has a value of less than or equal to 3.448, then it will be classified as cancer epithelial, and cancer epithelial can further be distinguished into either cancer epithelial if it is, or if it's not, then it gets classified as normal. This is a second confusion matrix that I was talking about to further refine the data. And here we can see that there's no misclassifications within the cancer group, as all 28 correctly classify as cancer, while as in the normal there is some misclassification. 2 of them misclassify as cancer, but 3 do classify correctly as normal. And this was the feature significance that I included to help show us the top features that were obtained from the models that I used. The top 10 features that are listed across the x-axis are correlated to the genes that I have listed here. These are not all the same genes that were obtained from the article, so further research would need to be done to either clarify or create a comprehensive list of which genes are most significant. So through these methods and analysis of the data, I've learned that while there is significance in the type of cell within the breast tissue, there are also other factors that contribute to distinguishing between a normal or cancerous cell. Those other factors will need to be further explored, as within this project, I focused on creating a better model for the cancer versus normal cells. Additionally, it was found that compared to the significant genes found by the differential gene expression model done by the authors, there are other genes based on my model that showed significance in the improved classification confusion matrix. This points us to the fact that there are a variety of genes that are affected within the tuber microenvironment, and with other machine learning analysis, we can refine the list of genes and find commonalities. Using and testing different algorithms is important so that we can compare the results that are yielded and continue finding more discoveries. Overall, the main takeaway is that while molecular analysis is not as common for gene expression of cancer cells, we're able to find significance in stromal cells and their presence in breast cancer. And hopefully with using more techniques and different ones, we can find models that have greatest accuracy. Overall, the prediction was supported by the results as based on my model, different genes resulted as the most significant compared to the ones found by the model from the authors in the article. This shows how various genes can be involved with the expression of breast cancer through epithelial and stromal cells in cancerous breast tissue. These findings will be helpful for science research, as these techniques can be used for other cancers and evaluating the effect of stromal cells in relation to disease progression. Thus, for further studies, Different algorithms can be tested on this data to dive further into the comparison of the cell and tissue types. Ultimately, the goal is to find the best technique that provides an accurate model to support the data and advance the predictions. Next steps would include working with these findings and continue to work with code that can further analyze the data to find better models and ultimately have better accuracy.